Okay, so my job is to introduce the next speaker, Dr. Judy Cameron. If you watched the PBS Frontline special by Dave Iverson, My Father, My Brother, and Me, you'll recognize this neuroscientist. Her work researching monkeys with Parkinson's symptoms, those that exercised and those that didn't exercise, was featured in that film. Dr. Cameron received her PhD in physiology from the University of Arizona. She's a professor of psychiatry at the University of Pittsburgh. That's my alma mater and hometown, by the way. She is also an affiliate senior scientist at the Oregon National Primate Research Center. Dr. Cameron is a neuroscientist and an expert on how the brain works. Her research focuses on the effects of everyday life stressors on long-term health. One area in particular that she researches is how exercise affects the brain. And of course, this is of great interest in the Parkinson community. Dr. Cameron is a member of several organizations related to the study of the brain, the MacArthur Foundation Research Network on Early Experience and Brain Development, the National Scientific Council on the Developmental Child, and also an organization called the Dana Alliances for Brain Initiatives. This organization is committed to advancing public awareness of the promise and progress of brain research. Today, Dr. Judy Cameron is speaking on neuroprotective effects of exercise in Parkinson's disease, from the lab to the clinic. Please pull out your yellow handouts. You can look at the objectives, the learning objectives, and you can follow along with her slides and take some notes if you like, and read a little bit more about her research. And please help me in welcoming Dr. Cameron to Richmond.
It's a great pleasure to be here, and the morning so far has been wonderful. I think I will follow very well after Dr. Wooten. You're going to learn about, can we do something about this progressive disease that you've just learned about? Oh, this works. Yeah, you can't put it on. Okay. They have me standing over here because I'm going to show you slides, and then I'm going to show you a little movie clip. Can everybody hear me? Yeah. Okay. So, no? Okay. Here. Yes. Here. Hang on. Okay. Can you hear me now? Yes. Okay. We're going to be talking about monkeys, and very frequently people start laughing, and I heard some laughs. Yes. People think, monkeys? Why, if you want to know about a complex human disease, would you be studying monkeys, for goodness sakes? There's plenty of people you could be studying. But I think you had a very nice introduction this morning to how complex this disease is. And different people live in different lifestyles. They're exposed to very different things in the environment. And so it becomes difficult to control lifestyle factors. That's what my laboratory studies. We study how do changes in lifestyle actually affect the way the brain works. And so that we can run very carefully controlled studies, we use monkeys, where we have the possibility of controlling factors a lot more than we do in most human studies. For example, we don't let them go out late at night and party, and we don't let them drink alcohol or eat a bad diet. So these monkeys live in a nice group social situation. They're inside, but they see the outside. There's skylights. There are no predators, no alcohol, no real stresses. And therefore, we can change things just a little bit. And what we're going to talk about today is changing how much exercise they get. And you're going to see that that's going to actually affect the brain. Now, I'm the person talking to you today, but this is a team effort because we're going to look at a lot of parts of the brain. And I like to start by introducing the people who are doing the work. In Pittsburgh, we work with Michael Zygmunt, and several of you asked me this morning, did I know Dr. Zygmunt? I am very, very lucky to know Dr. Zygmunt. I am a neuroscientist that looks at how does lifestyle affect your brain? How does exercise affect your brain? Dr. Zygmunt is studying Parkinson's disease, and he's studied Parkinson's disease for the last 30 years. And we teamed up together to do this work. However, it's very nice to be working with an expert on Parkinson's disease. We're going to look at the brain non-invasively by PET scanning so we can see those dopamine neurons that you heard about this morning. And we do that in collaboration with Brian Lepresti and Chet Mathis, who run the PET Scanning Center at the University of Pittsburgh, and with Rahana Leek. Zhiming Zhang is at the Morris Udall Center. The Udall Centers across the country fund Parkinson's disease research. He's a neurosurgeon at the University of Kentucky who developed the monkey model that we use. And he actually drives up to Pittsburgh every time we do these experiments and does them with us. So he's a very close collaborator. We want to know not only how does motor function change, how does movement change, and what's happening to the dopamine neurons in the brain, we want to know what else is going on. And the first talk did a beautiful job of telling you that's where the research is going to go. We want to know what else is going on. And we work with Karoli Mariniks at Vanderbilt University. He studies gene expression, and we're starting to identify all the different genes that are changed in Parkinson's models, and then with exercise. What can exercise do to those genes? We work with Bill Greeno at the University of Illinois. Bill is one of the first scientists who showed that exercise changes brain function. He's an anatomist, and he asks, where in the brain are there changes? So we are not only looking at dopamine neurons and Parkinson's symptoms, we're looking at many parts of the brain. And we're very fortunate to work with Nancy Williams at Penn State University. Now, she doesn't look at the brains of our monkeys. She actually studies people. She develops exercise programs for people. For the American Heart Association, exercise programs that will improve cardiovascular strength. For a lot of different foundations that are interested in using exercise. And she's developed a program for our monkeys that's very analogous to programs developed for people. So once we have our monkeys exercise using the exercise programs she's developed, we can say the same thing would happen in brains of people. So we're going to look at effects of exercise on a number of different processes, on how does exercise change gene expression in the brain, how does it change blood flow in the brain. Many people that I talk to think exercise doesn't affect the brain. Exercise affects your muscles, it affects your heart, it affects how you breathe, it affects your ability to control your body weight. But I have news for you, and you probably already could tell me this. This is a wonderful group to be talking to. You know exercise affects the brain. Exercise also affects two processes in the brain. The production of new nerve cells, or new brain cells, called neurogenesis. And this happens in a very specific part of the brain that helps make memories, called the hippocampus. Exercise, probably more importantly, affects the support cells that surround functioning brain cells, and these are called glial cells. Glial cells make all sorts of products. We're going to be concerned with protective products, and we'll show you that exercise increases the production of protective products from glial cells that then are neuroprotective and help protect your brain against insults. And at the end, then we'll talk about Parkinson's disease. So I'm going to start, the talk is divided into 3 sections. The first section is work we've done in collaboration with Bill Greeno and Karoli Mirnix to ask, okay, fairly globally, how does exercise affect the brain? And we started this study about 8 years ago with 3 groups of monkeys, 2 age groups, middle-aged monkeys and older monkeys. Now, monkeys live to be between 25 and 30 years of age, so the older group, 15 to 17, would be equivalent to about a 60-year-old person. And this is around the age that you start to see an increased incidence of Parkinson's disease. So we've been very interested in this age group of monkeys. But we also wanted to ask, do you get effects of exercise earlier? I often get asked the question, well, should we be encouraging people to exercise earlier? Yes, you should be encouraging people to exercise earlier. But the effects of exercise in a younger brain are not exactly the same as effects of exercise in an older brain. So there were 2 age groups. There were 3 experimental groups. There were runners. They ran in an exercise regimen that people run at. If you go to a gym, they say take your heart rate and try to get it into a target range for your age. And we used the exact same paradigm. So we had runners that ran for 5 months. They ran an hour a day, 5 days a week, which is what's recommended for improving cardiovascular function. We had sedentary animals. Now, it's very exciting to be a monkey and get to interact every day with us. So the sedentary animals got to get out of their group living situation and come to the treadmill room, but they just sat on the treadmills and they watched the runners run. They weren't so keen on that, but they did it. And we had run stops. These animals ran for 5 months and then they sat for 3 more months. And the reason we had that group is I get asked the question all the time, well, do I have to exercise always? Do I actually have to keep this up? Can't I just exercise some and then take a break? Well, we wanted to ask that question. Can they exercise for 5 months? Do they still have advantages 3 months later? And you'll see there are some advantages, but you lose a lot. It would be a lot better to keep exercising. We asked not only how did they move, but how alert were they? And we can get monkeys to do pretty complex learning paradigms, and it's a simple situation. We bring the animal in and we put it in a cage that has open panels. They can reach their hand through and do things. And there's a tray in front of them with little wells, and we put a treat in the well. Now, some animals like little pieces of apple or a little piece of orange. Other animals like M&Ms or jelly beans. So we find out what they like. We put it in the well. We put a toy on top. Then they have to move the toy and get the treat. And you can do lots of tests with this setup. You can have them have to move the toys in different orders, or you can pull a screen down and then bring it back up and see if they remember which toy the treat was under. So that's what that's showing. And what we found is, if you look in the top panel here, the animals that exercised, which are shown, the runners are shown in the black bars, learned to use the testing apparatus much faster than the other animals. So the sedentary animals are in the open bars, the runners are in the dark ones. You see, the dark bar took only 4 days on average to learn to do the test. The sedentary animals took a week to learn to do the test. Why is that? Well, you can see that the runners were more alert, they were more attentive, and they were doing a lot more trials. They didn't make fewer mistakes. They made mistakes at the same rate. So a scientist would say they weren't smarter. But we can ask, do you think it makes a difference to be more alert, more attentive? You learn a lot faster if you're more alert and more attentive. It's a lot different than being droopy. Okay, what else was going on in the brain? We're going to look at some of the things that Bill Greeno found out in this study. In each of these figures, you're looking at the middle-aged animals on the left and the older animals on the right, and a picture is shown on the far left. We found that exercise increased blood vessels in the brain, and a picture of blood vessels is shown over on the left. You can see that in the older animals, the running group, the dark bar, is taller than the sedentary group, the white bar, or the run stops. If you are exercising, you have more blood going to your brain. What does blood carry? It carries oxygen, it carries nutrients. So you get more nutrients, more oxygen to the brain in running individuals. And that's happening even when they're not running. They have more blood vessels. Now, the sad thing is they go away. If you stop for 3 months and you don't exercise at all, oh dear, not as many blood vessels anymore. You don't get the same effect in middle-aged animals. So as you get older, your brain becomes more susceptible to exercise. It's good to exercise as you get older. Turns out it's good to exercise when you're middle-aged too, but it does different things. Here we asked, does it create new brain cells, new neurons? And you see that exercise increased new brain cells in middle-aged individuals, and those actually last. They don't stop and go away when exercise ends. So exercising when you're middle-aged has a very positive effect, and it's an effect that lasts. The brain is less able to make new brain cells as you get older, so you're dependent on the positive effects coming from an increase in blood flow. And the last thing we looked at is, does exercise affect the rate of production of those support cells, the glial cells? And the answer is, it does, and it does no matter what your age is. And the effect lasts. After you stop exercising, you still have more glial cells. So exercise is doing a lot of good things. So what did we conclude from this work? We concluded that exercise increased blood flow, it increased production of new neurons, especially in parts of the brain, the hippocampus, that are involved in memory, and it increased the production of support cells. The clinical implications are that exercise might counteract effects in the brain that occur with aging, and in particular, neurodegenerative diseases like Parkinson's disease. So what we are doing now, based on this research, is we would like to know, how does exercise affect gene expression in the brain? And I'll show you a little bit of that data. We'd like to know, can exercise actually slow the rate at which your brain ages? And we have a whole series of studies looking at that. And the question I get asked a huge amount of the time is, how much exercise does it take? And what type of exercise? And how long do the effects of exercise last? I had somebody at a talk I gave recently ask me, would Pilates help? And joking, I said, we have a Pilates instructor for the monkeys, and we'll be able to answer that question. No, we don't have a Pilates instructor for the monkeys. There are lots of questions that are going to be much better answered by looking at what exercises do in people. And I think all of you will be doing those experiments. Okay, I'm going to show you the second set of data that we'll talk about today. And this is work done again with Jiming Zhang and Michael Zigman, experts on Parkinson's disease, and the people doing PET scanning, as well as the effects on gene expression. Parkinson's disease, as you learned in a very, very nice talk today by Dr. Wooten, is something that happens, and he talked about the bracts, and this is in fact a diagram from their publication showing that it looks like it's an ascending disease. There are problems occurring in many brain regions, but they eventually get up to the dopamine neurons, which are shown on the right side. Normally, the substantia nigra, shown by those very dark cells, is very marked. But with Parkinson's disease, there's a loss of dopamine neurons, and you have much less darkness in the substantia nigra. You have a loss of dopamine cells. What we're going to be asking is, can exercise prevent the loss? So, this is a graph that really shows what Dr. Wooten very beautifully talked you through. We have percent of dopamine in the part of the brain that controls motor movement, and we're looking over age. And the yellow line shows you that slowly as you age, you lose dopamine function. That's a normal characteristic of aging. But you don't develop motor problems because you can lose a lot of dopamine before motor problems develop. He gave you a number of about 60 to 70% of dopamine neurons can deteriorate, and you still have fairly good motor function. However, if you have either a genetic predisposition to lose dopamine neurons faster or dopamine function faster, or you're exposed to a chemical in the environment, and there are many chemicals. There are pesticides. There are chemicals found in chemical warfare. There's chemicals found in heavy industry, all of which affect dopamine neurons. So you can have a number of life exposures. Shown by the red line, you start to see a deterioration of dopamine neurons. And once you get past where the green dashed line is, you have motor dysfunction. So in this study, we tried to mimic this in a monkey model. We used older female monkeys. Remember, we looked at effects of exercise in the brain, and they were dependent on age. So in this case, we used older monkeys. We used a running regimen. They were either running at 80% max heart rate or a little bit slower. Now, why did we have two groups of animals, a faster group of runners and a slower group? Well, the reason was we have a number of colleagues that are neurologists, and they treat people with Parkinson's disease, and they were concerned that maybe people with early stage Parkinson's disease wouldn't be able to run as fast as we were recommending. So let's have a middle group that's joggers. They're not running as fast. And that made sense to us. Now, since we did this study, I've met a lot of groups of people with Parkinson's disease, and I have to say, they tend to be very, very motivated. And if there's a group that's motivated to exercise, this is the group. So very frequently, they can run plenty hard, and that isn't so much an issue. But you'll see that we saw effects no matter whether the animals exercised a little bit or exercised more. The effects were dependent on how active they were, so it's always good to exercise more. We used a drug to create the Parkinson's symptoms, and this is MPTP. The frozen addicts, which first were found in San Francisco at a time where I actually happened to be in college at the University of California at Berkeley. So I was right there in the Bay Area when the frozen addicts were first occurring. They had injected themselves. They were young kids, 18 to 20 years old, who'd injected themselves with a street drug, MPTP, and it caused a loss of dopamine neurons and Parkinson's disease. So we give the monkeys a very, very small dose of MPTP, and we do it just on one side, just on the right side, and we get a little bit of loss of dopamine neurons in the right side of the brain. That way we can compare how they're functioning on the side that was affected with the other side of their brain. We're going to look at behavior, how well they do motor function. We're going to look by PET scanning, and you'll see pictures of the brain. And then we'll look at brain analysis. So this is a different way of looking at the experimental design. We take these aging female monkeys. We're going to have them run, jog, or be sedentary for a 3-month period of time. We're then going to give them the dopamine toxin, MPTP, and we're going to watch them for the next period of time. What we're going to look at here is the results of the PET scanning. So this is a sedentary animal, and on the right side of the brain, you see there's a loss of dopamine, which is shown by the orange-red color. That's high levels of dopamine in the substantia nigra and the striatum on the left side, but they had the MPTP on the right side, and there's a loss of dopamine. So you're looking at the striatum made up of the caudate and putamen. This is an exercising animal that was jogging. And what you see is that on the right side of the brain, there's more dopamine now. They jogged for 3 months before they got the toxin. We think what's happening is the exercise is protecting those dopamine neurons so they don't get as damaged when they get exposed to the toxin. And then if they were exercising and running at 80% max heart rate, you see almost no loss of dopamine. The exercise has done a very good job of protecting the dopamine neurons. So it's reducing the deficit that we see. We also looked at motor function. And you heard this morning, you can look at how rigid an individual is or how slow. So you're looking at those two symptoms. The sedentary animals are shown in the closed circles, the black circles, and they showed quite a bit of rigidity after MPTP. The jogger is shown in the open circles, and there was a little bit of rigidity, but it was much less. And actually, I've got that backwards. The runner was the triangles, and showed no rigidity. The sedentary animal is the circles, and it showed a lot of rigidity. And the jogger showed intermediate rigidity and only occasionally. If we look at bradykinesia, slowness of movement, we see the same thing. The sedentary animal, that top line in the closed circles, showed a lot of slowness. The runners, both runners, showed no slowness at all. We then used a more sophisticated way of looking at motor function, and this is called a monkey movement assessment panel. Some of you may have used something like this. They now make these for people. And you put your hand in through a slot, and then you do a motor task. In this case, there was a platform, and the monkey had to get the treat off the platform. There's an infrared beam, so that when they put their hand in, it breaks the infrared beam, and you can time exactly how long does it take to get the treat. So it's a very accurate way of looking at motor function. And what we see is the sedentary animal wouldn't use the left hand at all. If you damage the right side dopamine neurons, the motor pathways cross, and you don't use your left hand. So the slowness, the rigidity from a right side damage is in the left side of the body. The sedentary animals wouldn't do this with their left hand. The jogger, shown in the middle, was slower with the left hand than the right hand. The left hand is shown in the dark circles, the right hand is shown in the open circles. So there was a little bit of impairment, but not a lot of impairment. There's not much difference between those lines. And the runner was completely protected. If you look at the first set, it could use the right and left hands the same. So we made the task harder. We made them thread a lifesaver off of a rod, and the runner could still do it equally with the right hand and the left hand. The runner was very, very protected as far as motor function was concerned. Next, we looked in the brain, and what we're looking at is different chemicals in the brain that are in the dopamine pathway. The TH right at the top is tyrosine hydroxylase. It is the enzyme that makes dopamine. And what you can see is that the sedentary animal on the side of the brain, sedentary is shown to the far left, on the side of the brain that got MPTP, there's no band of protein for tyrosine hydroxylase. That animal had lost almost all their dopamine neurons. If you look at the 60% runner, the one in the middle, you can see that they lost a lot of dopamine neurons also. But the 80% runner had almost all its dopamine neurons still. So we've shown you by motor function, by PET, and by chemistry that exercise protected those dopamine neurons. How is exercise acting? How do you get effects of exercise on the brain? Well, we think that the mechanisms are the effects on production of brain cells, the effects on production of neuroprotective factors, and the effects on new neurons. And this is work we're doing right now in collaboration with Michael Zygmunt at the University of Pittsburgh. We think exercise is protecting the brain by producing new neurotrophic factors, factors the brain makes that protect neurons. GDNF is one of these neuroprotective factors, but there are a number of them, and they affect signals inside the cell. This is a schematic diagram of a dopamine cell, and we're looking at the signaling pathway that's turned on by exercise that seems to counteract the effects of cellular stress. So cellular stress may be something you inherit. It may be an environmental toxin, and it causes damage to cells. We think exercise turns on protective factors that counteract that damage. So we conclude from this that exercise protects dopamine neurons, and we're fairly sure it will protect against things like Parkinson's disease and the progression of Parkinson's disease. I'm going to show you the clip from Frontline to reinforce this, and then after that, I'm going to show you a new analysis of this data. This is work that came out of the lab in the last couple months, and it will have a huge impact in how you think about exercise.
Or to put it the other way around, would more exercise mean less Parkinson's? The answer to that question may come from a surprising source.
We're probably the only lab in the world that runs monkeys on treadmills, so it is not an everyday occurrence.
To test the impact of exercise on Parkinson's, the University of Pittsburgh's Dr. Judy Cameron has divided monkeys into two groups, the runners and the watchers. The running monkeys train for 3 months. The watchers, well, they just watch. And then both groups are injected with the toxin MPTP, the same toxic substance that caused instant Parkinson's in the frozen addicts.
What we're seeing is monkeys that have run on the treadmill have very little effect of the MPTP. And they're still able to use the side in which they received the injection, and they can use it very normally. The monkeys that are running look very different from the sedentary controls. The sedentary controls aren't really using their arm. They're letting their arm hang down. It makes us think that exercise somehow changes how the brain is functioning and protects it against damage.
Everybody ready?
Ready.
To see exactly what's going on, the animals are then given a brain scan. The results prove to be dramatic.
It's a pretty massive lesion.
The MPTP injection has decimated dopamine production in the monkeys who watched but didn't run.
But in contrast, the running animal,
The lesion is much less.
In fact, in some of these slices, it'd be hard-pressed to know that there was a lesion at all.
Right. So
In fact, while the MPTP knocked out dopamine production on one side of the brain in the sedentary monkeys, the running monkeys were only minimally affected. Their dopamine production remained virtually normal.
So what we're finding is that when we exercise the monkeys on the treadmill, for the amount of exercise that clinicians would recommend that middle-aged people undertake, their brain is less likely to be damaged when there's an insult.
And that's the key question. Is what's good for the monkey good for you?
Okay, so one of the main questions I get asked is, how much exercise? What kind of exercise? We really want to beat Parkinson's disease. We want to be better. What exactly do we have to do? And we would love to answer that question. I think people think of basic scientists of being people that are in their lab coats, locked away in their labs, that don't care about the issues that people care about. But that's not true. We care a lot about these issues. And what I'm going to show you next is that you probably don't need to exercise per se. You need to be active. And this is new data collected over the last year and analyzed in the last several months. It's work done by one of my graduate students, Eleanor Sullivan, and Karoli Marnix, looking at effects on gene expression. So for this part of the study, we had monkeys wear activity monitors. You could wear a pedometer. Monkeys don't walk back and forth in a nice line or stay on the ground. They jump, they climb, they go in all directions. And so we needed an accelerometer. It's like a pedometer, but it measures activity in every direction. And they wore it, it's about the size of a quarter, in a little neck band that they wear around their neck. This is loose fitting. It doesn't bother them at all. Once they get used to it, they wear the activity monitor all the time. What we can see, what you're looking at here, is activity across the 24-hour day in two different monkeys. On the left side, you see a less active monkey. You might refer to people like this as a couch potato, somebody who really doesn't do much at all. So right in the middle of that graph is the 12 hours of daytime, and this monkey moves very, very little. Each excursion up is going to be a movement. This monkey isn't moving much at all. They stay still. And then at night, when they're sleeping, they don't move much either. On the other hand, you have a very active monkey on the right side. This animal, when it's sleeping, isn't moving, but the minute it wakes up, it's all over the place. And it moves a great deal. It moves up to 8 times as much as the sedentary monkey. Now, an 8-fold difference in something is a huge difference. If I said, I have high blood pressure, you have low blood pressure, I would mean there was just maybe a 20% difference, not even a 2-fold difference. This is an 8-fold difference. Some individuals are very, very active. They're moving all the time. Other individuals just don't move at all. So monkeys are a lot like people. There's sedentary monkeys, there's active monkeys. With Karoli-Marinick's, we asked, does activity change what genes your brain expresses? And I'll walk you through this picture. It might look complicated to start with, but we're looking at expression of about 100 different genes. That's the list there. You should memorize it. A list of 100 different genes in the part of the brain where the dopamine neurons are that lead to Parkinson's disease. And the animals have been arranged. There's 14 animals in this study. So the least active animals on the far left, the most active animal is on the far right. And that green triangle shows you least active to most active. And you can see that you're looking at genes. The red color shows high levels of expression. All the genes at the top, the first half of the list of genes, are expressed at very high levels when you're sedentary, and the more active the individual is, the lower the expression of those genes. Activity is inhibiting those genes. On the other hand, all the genes at the bottom of the list are just the opposite. They're expressed very low levels, shown by the blue color, when an individual is sedentary. But the more active the individual is, the more the gene is expressed. So this is one little region in the brain. There are 100 genes that are influenced by how active you are. When you ask the question, how is exercise affecting your brain? It's having huge effects on your brain. This is just where your dopamine neurons are. Remember, I told you that activity would also influence memory. If you look at the part of the brain where memory is, there's about 200 genes that are affected, and many of the genes are different from these genes. Activity is having a very big effect on the brain. It's turning on some genes, it's turning off other genes. We decided to reanalyze the data I just showed you. We didn't collect new data. Those animals had been wearing activity monitors also, and we reanalyzed the data. So you're looking at the PET scans you're used to looking at now, and you're looking at a plot. You're looking at the PET scan information versus how active they are. And you can see there was a really, really strong correlation between activity level and how protected the brain was. As you move up this line, the animals become more active and they are much more protected. It's hard, I think, for you to see the symbols on this graph, but the symbols are, were they sedentary animals, were they running, or were they jogging on the treadmill? The bottom line is, it mattered how active they were. It didn't matter what exercise group they were in. If they were active, which is going to be a combination of are they intrinsically active and are they exercising? The exercising animals tended to be the more active animals because exercise is a big dose of activity. If you exercise every day, overall, your daily average of being active is higher. So exercise increases the chances that you're active. But what probably matters to your brain cells is that you're active. This shows you the same information, and now you're looking at brain chemical levels, levels of that enzyme that makes dopamine. The same story is true. It doesn't matter what exercise group you're in, it matters how active you are. So, what does that mean? Well, I think that's really, really good news. It means that being more active, regardless of how much aerobic exercise you do, can protect dopamine neurons. And we think that this has very big clinical implications. It means that maintaining an active lifestyle may significantly protect the brain against neurodegenerative diseases, Parkinson's disease, Huntington's disease, but there's a lot of work on other neurodegenerative diseases, Alzheimer's, or neurodegenerative events like stroke. It appears that exercise is protective for all of these neurodegenerative diseases. It's useful in decreasing the rate of progression of Parkinson's disease. The really nice news is this is a health strategy that is accessible to many people regardless of their baseline fitness level. So if you can't go out and do aerobic exercise, just do something. Be a little more active. Okay, thank you very much.